here. They're on the front. Please come up and get one. Help yourself to that. And I'll remind you that note cards must be 100% handwritten, so no printed cards other than you're printing by your hand. And any figures on there must be drawn by your hand, okay? So make sure that you do that. You will have to turn the card in, so yes, I will know. And that means that your card needs to have your name on it. So please make sure that your card has your name on it so that we can track that, okay? So you need to turn the card in, you need to have your name on it, and as I said, you need to turn a card in. So if, oh my God, I forgot my card, et cetera, then I'd say you probably better let me know before the exam starts, right? Okay, so that's important. The exam itself is on Wednesday. You guys don't want to have a Monday? I told you it'd be a surprise today. Okay? The exam is on Wednesday at 6 p.m. in this room. And as before, please position yourselves as I've asked you to sit appropriately. And that helps us to get started. I haven't written the exam yet, but the exam will be about 150 points. It may be a little bit shuffling of the percentages of the long and short answer and so forth. So, but I will keep the same basic format. You will see a short answer, you will see a problem section, and you will see a longer answer section. Okay? I don't think time should be an issue. I really don't. I'll give you a full hour and 50 minutes to complete your exam, and you should be set with that. Review session. I have scheduled a review session, and it will be on Saturday at 1:30 p.m. in ALS 4001. Now, there will probably be another group in there before us doing a review session, so be polite. If you see people in there talking, don't go and sit down and enter the room. Please wait for them to finish. They will be done by 1:30. And that other group, some of you are in that class here, that is by 314, has some sort of review in there at that time. Okay. Let's see, those are the main announcements that I have. Yes, Cassandra. Am I going to give you a choice for the long answer on the final? I'm inclined to say probably not, but I haven't decided. I did have some hassles with the last one. It's always the problem, people, that ruin it for everybody else. And I don't like fighting those battles. I really don't. Well, if they can't follow directions, but then, you know, the point is they end up being mad, so if I don't do any of that, then nobody's mad, right? Except now they're mad at me. Well, I will consider giving you a choice, okay? But I will warn you, we will absolutely enforce that rule as I've done before, okay? That needs to be done. But I'm not saying yes or no. I haven't decided. Okay. Other questions? I will remind you that I think probably the best way to study for the exam is to use the 2 exams that you've had so far as an outline to the previous material. The 2 exams that you've had so far constitute what will be a total of about 75% of the final, and the new material will be roughly 25% of the final. And yes, I do try to break things down proportionally to how much I've covered them in the class. So do that. Look at the old exams. You're not going to see the same questions with the possibility of 1 or 2 minor exceptions. You're not going to see the same questions. You will see the same topics. And I also recommend looking through the highlights. I think highlights, as I said before, are my view of what I talked about. And since what I talked about is the highest priority, I think the highlights are a good place to start, get things going. Okay, now, we are going to finish glycogen metabolism today. I have a couple of surprises for you, and we'll see how that goes as we get going along. So first, you've seen that glycogen metabolism regulation is fairly complex. And I really want you to sit down and flesh it out. You can flesh it out on your note card. That's fine too. Okay? But it's important that you understand what's happening in that regulation. I've had a couple people ask, well, am I going to ask you a pathway to just regurgitate like that? And as you've seen on my exams, I tend not to do that. I do tend to pick out things out of pathways that are important. I didn't ask you, for example, to draw glycolysis. Okay? But I did ask you questions from glycolysis. So those are fair game. Okay. So I came back to this figure because I really had the sense last time when I was talking about it that people were sort of perplexed, probably tired, probably not really understanding what was going on. So I thought I would say a few words about it. Then I'm going to come to what I call the curveball about glycogen metabolism. And glycogen metabolism is pretty straightforward in many respects, but there's a curveball that is really interesting, I think, in terms of how it works and what it does. First, what we're looking at here is the regulation of phosphoproteinphosphatase. Phosphoproteinphosphatase is the enzyme that takes the phosphates off of the proteins that have them. So phosphoproteinphosphatase will take phosphates off of, for example, glycogen phosphorylase A, glycogen synthase B, okay, phosphorylase kinase, all of these, okay? And it will also take it off of its own inhibitor when it's given a chance. Now, how does that work? Well, let's start over here on the inactive side, which is something different from what I talked about before. Last time I talked about how we turned the phosphatase off. What I want to do is just say a couple words about how we turn the phosphatase on, and then I'll repeat how we turn the phosphatase off. How do we turn the phosphatase on? That's actually a fairly complicated pathway, and it's one that I'm going to spare you. We're not going to talk in detail about how we turn the phosphatase on. Okay? But suffice it to say that there are other phosphatases present in the cell, and these other phosphatases in the cell can be activated as a result of action of the insulin receptor. So when the insulin receptor is activated, the other phosphatases can remove a phosphate right here off of the inhibitor. Okay? Removal of one of these leaves a little bit of phosphoprotein phosphatase active, which can in turn start taking phosphates off of everything, including other inhibitors that have a phosphate on them. So once the insulin receptor gets activated, we see activation of a lot of phosphatase action in the cell. And that phosphatase action in the cell is ultimately going to result in this guy becoming fully active. So we see here that the phosphatase loses its inhibitor. We get a somewhat active phosphoprotein phosphatase. That somewhat active phosphoprotein phosphatase can take the phosphate off of this G and then give the phosphoprotein phosphatase a place to bind. And of course, when it binds to the G, now it is fully active and able to go out and start taking phosphates off of everything. Okay? On the other hand, if we want to stop the phosphatase from acting, it requires action of the other hormone receptor, the other hormone receptor being the epinephrine or glucagon receptor. So to stop the phosphatase from being active, the kinase cascade that results in the activation of protein kinase A kicks in. That cascade causes a phosphate to be put onto the G, which causes the PP1 to be released. That's what you see here. PP1 is released. Notice the inhibitor in this case is shown with no phosphate, and it's not bound to the PP1. So when the phosphate's off, it's not bound. The addition of the phosphate by protein kinase A to the G causes PP1 to be released. It is now less active than it was before. The protein kinase A also phosphorylates the inhibitor, and when it phosphorylates the inhibitor binds to the PP1 and causes the PP1 to become completely inactive. Okay? So as a result of this, the signaling through the epinephrine pathway will inactivate PP1. Signaling through the insulin pathway will activate PP1. Okay? And again, we're seeing reciprocal effects, exactly opposite effects, depending upon which receptor is getting hit. Okay, does that make sense? Keep in mind that when we activate the phosphatase, what we're going to do is we're going to activate glycogen synthase. We're going to inactivate glycogen phosphorylase. Okay, questions on that? Laurie?
When PP1 is in the less active phase, it can dephosphorylate its inhibitor, GM protein, or anything else.
Or anything else, that's correct. So it can start that dephosphorylation process. Yes, Hong? What did I say about what? The protein kinase A phosphorylates the inhibitor. So it phosphorylates both the G and also the inhibitor. Notice I'm not calling this a G protein, even though I really want to. It's different from a G protein. It is a protein called G. It's kind of like the artist formerly known as Prince, right? The protein also known as G. I should have a little symbol for it or something. It'd be kind of cool. Okay. Other questions? You're a quiet group today. Maybe it'd be a good time for a joke, I think. One of the surprises is I have a joke, and that's not always a surprise, but in combination with other surprises, that's unusual. So I really like this joke, okay? And if you know the answer to it, please don't yell it out. I like that joy myself. What's the difference between a golf ball and an SUV? Tiger Woods can drive a golf ball 300 yards. I love that joke. Good old Tiger. Okay, as if that was really news or something. Okay. Now, here's a figure that confuses people, and I'm going to show it to you. I'm even going to talk about it briefly, and I'm not going to expect you to know it. And the reason is because it's confusing is it's an insulin signaling pathway that's actually activating some kinases. And we're thinking about insulin receptor in terms of phosphatases, so this would seem counter to that. And it turns out that this kinase, a separate kind of kinase cascade, is responsible for activating some of the phosphatases. So it's confusing, all right? So don't sweat this, all right? Don't sweat this. The point of the matter is the insulin receptor can activate an awful lot of things, and as a result of this can cause other things to happen in the cell, ultimately that which includes phosphoproteinphosphatase becoming activated, okay? So don't sweat this. You knew that the insulin receptor was in fact a kinase because that was one of the questions on the last exam. And it's a tyrosine, a protein tyrosine kinase, and so not surprisingly, it is in fact going to put phosphates on other proteins. The ultimate result of this is activation of a phosphatase, however. Okay. Now, it's time for the curveball. And the curveball is that glycogen metabolism that I've talked about so far is complicated, all right? There's no question that it's complicated, but there is a nice logic to it. And the curveball actually has logic to it too, but I think it's kind of odd. And I'm going to show you the experiment that reveals the oddity to you in a second. But the beauty of glycogen metabolism to this point has been that it's very nicely reciprocally regulated. We see the phosphorylase being activated by things that turn off the synthase. We see the synthase activated by things that turn off the phosphorylase and vice versa. So this is nice and it's balanced and it's simple. And we're not going to upset that balance. But there's something that's kind of surprising about glycogen phosphorylase, and I want to say a word about that. The surprising thing about glycogen phosphorylase is actually revealed in this experiment. This experiment is something that people did where they had purified glycogen phosphorylase in the A form, and they had purified glycogen synthase in the B form. You recall that both of these are the forms that have the phosphates on. Okay? So they had glycogen phosphorylase in the A form and glycogen synthase in the B form. And when you measure the activity of them, not surprisingly, the glycogen phosphorylase is very active, and the glycogen synthase is not very active because we think about the B form as being the much less active form. Then something interesting. The something interesting is you simply add glucose to the tube. Now I want to emphasize, excuse me, here, that we're not talking about cells. We're talking about in a test tube. So we've got purified glycogen phosphorylase A, and we've got purified glycogen synthase B. All right? So this isn't something where there's a hormonal system that can activate things or change things. We simply have two enzymes present in the cell. All right? So we don't have cellular signaling. We don't have all these other possibilities there. Okay? Make sense? When we add glucose, something interesting happens. Glycogen phosphorylase A drops fairly quickly in its activity. Fairly quickly it drops its activity just by the addition of glucose. And then slowly, we see glycogen phosphorylase B gaining its activity. And what's happening when people studied this was they discovered that glycogen phosphorylase A was flipping into the B form, and glycogen synthase B was flipping into the A form. Now, how could that happen? Based on what we talked about in class, what would it take for that to happen? What's that? More than one substrate, she says. No, not quite. More than one subunit. Okay, more than one subunit is correct. Okay. What's the other subunit? What enzyme activity has to be present? What does it take to convert glycogen phosphorylase A into B? It takes a phosphatase. And what does it take to turn glycogen synthase B into A? The same thing. It takes a phosphatase, right? This revealed something very cool. And this is the curveball. The curveball is that glycogen phosphorylase carries the phosphatase. It carries the very thing that will turn it off. Now, this happens in the liver, which is why you see this G called L here. L stands for liver. The one you saw before was M, and I said that there was an L and there's an M form. Okay? So this is happening in the liver, and it's in the liver that this is relevant because why? Why do you suppose this would be relevant for the liver? I saw you put a leg up. Is that it? It's funny. I thought it was your hand going up. I thought he's raising his hand, but actually he's raising his leg. I gather that didn't mean that you knew the answer to the question. Why would this be important for the liver? Think of the experiment I just did. What was the experiment I just did? I added glucose. What does the liver have? Glucose from gluconeogenesis, right? Okay. So it's important that the liver be able to sense how much glucose is present and that the liver be able to activate a phosphatase when the glucose level gets high. Okay? It's important that that happen. Now, let's look at the actual way this occurs. So we start out with glycogen phosphorylase A. It's in the R state, which means it's the most active form. And this glycogen phosphorylase A is carrying around with it a phosphoproteinphosphatase linked to G. Okay? So when this guy is going around breaking down glycogen, it's carrying with it the thing that will actually inactivate it. In this form, PP1 is inactive. Okay? I didn't say that before. It's inactive. Before, when you saw PP1 linked to G, it was active. It would be active except for the fact that glycogen phosphorylase A is covering up its activity site. So PP1 is sitting here unable to act. It's being carried around like a spare tire by glycogen phosphorylase A. Okay? So glycogen phosphorylase A can be breaking down all the glycogen that it needs to. When it breaks down glycogen, it's of course going to make glucose 1-phosphate. Glucose 1-phosphate will get converted to glucose 6-phosphate. And glucose 6-phosphate, if we're in the liver, is going to be converted into glucose, right? As I've described in class, if the muscles that were using the glucose in the Cori cycle stop exercising, they stop taking up glucose. When they stop taking up glucose, blood glucose levels start to rise. When blood glucose levels start to rise, the liver stops pushing out glucose, and liver glucose levels rise. This means, therefore, that when liver glucose levels rise, what we see is that this guy gets converted into the T state. So glycogen phosphorylase A goes from the R state to the T state, and in the T state, it lets go of the phosphatase. Now, this phosphatase, which was previously inactive, is active like crazy. It's fully active. What does it do? It starts dephosphorylating everything. It takes the phosphates off of glycogen phosphorylase A, which helps convert A into the B. It takes phosphates off of phosphorylase kinase, so it becomes inactive. It takes phosphate off of glycogen synthase B, so it becomes glycogen synthase A, which makes it active. And we've instantaneously reversed the process. Now remember the power of enzymes. enzymes can work so rapidly that control of them is very important. Having something that is literally a hair trigger on the control is very, very useful here. Because once the liver glucose levels start rising, then we don't want to be breaking down glycogen. We want to stop it immediately. Otherwise, we're going to destroy all of our glycogen because enzymes work fast. Make sense? You're a very quiet group today. I should have saved the joke for later. Okay? Makes sense? All right. So this explains what happened in that experiment. The simple addition of glucose caused glycogen phosphorylase A to flip from the R state into the T state. That caused release of the G with the phosphoprotein phosphatase. This becomes active, it starts dephosphorylating things, and everything goes as we've seen before. So that was how we could have a purified glycogen phosphorylase A and a glycogen synthase B, and all we add is glucose. And what we didn't realize was that there were other subunits there, as Laurie said. The other subunits were the phosphoprotein phosphatase. Okay. Questions about that? Yeah?
So what specifically is glucose doing to deactivate? So glucose is what causes it.
Right. Okay. If you remember back when I first talked about glycogen phosphorylase, remember I said that we had glycogen phosphorylase glycogen phosphorylase A, and there was one thing that would flip it back into the T state? It was glucose. This is how and why. Okay? What does it take to flip from the T back into the R state? Nada. Nothing. Right? So as long as once the glucose disappears, then it's going to flip back into the R state. However, if we have taken the phosphate off, which is what's happening here, when it flips into the R state, it's going to be in the glycogen phosphorylase B, and that takes different allosteric effectors. Okay? So the control is very, very finely tuned, and people talk about this control that you see here as being a glucose sensor in the liver. It gives a very, very fine measure of the level of glucose that's there and is activating either glycogen synthesis or glycogen breakdown as appropriate. Notice that glycogen synthesis is of course going to use that glucose to remake glycogen. A pretty efficient system. It's recycling at its best, I think. Okay, other questions about that? The end is near. We're almost there. Okay. What's that? The end is what? The end is nigh. I think the end is night. No. It's dark in here. It will be dark in here on Wednesday night, but it's not dark here now. Okay. Glycogen. So the last things I'm going to talk about here in class relate to anomalies in glycogen metabolism. Okay. Glycogen, as I shown before, you can actually see it in an electron microscope, and you can see in this case a lysosome which is loaded with glycogen granules. Okay? This cell's got some problems in glycogen metabolism. And there are several diseases known that involve enzymes either directly in glycogen metabolism or indirectly in glycogen metabolism that affect glycogen metabolism that have effects that range from severe to surprisingly not so severe. And we're going to analyze one of those not so severe in just a second. Okay? So I'm going to show you a table. I'm not going to ask you to memorize the table. I'm just going to show you the table to give you some ideas of things that are there. Okay? It's a little hard to see. I know it's a small figure. But listed on the left are 8 different glycogen storage diseases that are there. And some of these have enzymes that are directly involved in glycogen metabolism, as I said. These include things like branching enzyme, debranching enzyme, etc. And others that are peripherally related, like glucose 6-phosphatase. Glucose 6-phosphatase, where would you expect to see that? What organ? Liver, right? For gluconeogenesis, okay? So this isn't a glycogen enzyme, but it's related because if we think about it, this is what's making glucose. And if we take away the ability of the liver to make glucose, what happens? What do we see? Well, we see, first of all, that there's an increased amount of glycogen in the liver. Is that what you would expect? Does that seem reasonable or not? Okay, why would that be something that you might expect to increase the level of glycogen? What's going to increase if I increase the concentration of glucose 6-phosphate? can't hear you. Well, glucose, no, because glucose 6-phosphatase is what's going to make glucose. So we're not going to see glucose. What are we going to see? Glucose 1-phosphate, right? Remember phosphoglucomutase? It's going to convert glucose 6-phosphate backwards into glucose 1-phosphate. And what's glucose 1-phosphate going to make? Glycogen. You got to have these in your head, folks. It's going to happen on the exam. Okay. Glucose 1-phosphate is a precursor of glycogen. So we would expect if we couldn't make glucose, what's going to happen is you're just going to keep making glycogen. It's going to get bigger and bigger and bigger. That's why you see enlargement. Okay. This is, notice it's very severe, failure to thrive, severe hypoglycemia, ketosis, blah, blah, blah. You can imagine that if you can't do gluconeogenesis, that you would have severe problems with Cori cycle, okay, among other things. So this is a pretty nasty disease. Pompe disease, alpha-1,4-glucosidase. And by the way, these are just for fun. These aren't, you're not responsible for these. Alpha-1,4-glucosidase, okay. This is missing the enzyme that's a portion of the debranching enzyme. What you see is a massive increase in the amount. You have a normal structure. Okay? Why would that be consistent with a deficiency in debranching enzyme? What's debranching enzyme going to do? It's going to take the branches away. Okay? So it means that if we can't take the branches away, when the phosphorylase is active, it's going to get to a certain point. It's not going to be able to go any further. So the branches, once they've been made, are going to be there. They're going to stay there. They're not going to disappear. Okay? So we're going to see, again, a massive increase in amount. We're not going to be able to go through the branching. And it's whenever we build new glycogen, it's going to keep adding more and more branches, none of which are going to be able to be removed. So, not surprising. Cardiorespiratory failure causes death, usually before age 2. Okay? Nasty. Cori disease. There's a name for the people who did the Cori cycle. Cori disease. Lacking the debranching enzyme. Like type 1, but it has a milder course. The one I want to analyze with you is McArdle's disease. McArdle's disease is interesting. Most of these are fairly severe. Okay? Liver failure causes death, usually before age 2. Death before age 2. McArdle disease involves a defective glycogen phosphorylase. Now, of all these, wouldn't you think that a defective glycogen phosphorylase would be a pretty nasty disease? In this particular disease, the glycogen phosphorylase is lacking in the muscle cells. It's present in other organs, in the liver, for example, but it's not present in the muscle. Muscle is needed, of course, to break down glycogen, give you a quick release of glucose. Look at the glycogen. It's moderately increased in amount. Not surprising, because you're not going to be breaking it down very well, right? Limited ability to perform strenuous exercise because of painful muscle cramps. Otherwise, patient is normal and well developed. You could have, there could be people in here, for example, who could have McArdle's disease and would probably, as long as they weren't trying to exert themselves very heavily, would have a fairly normal life. How is that possible? If I take off and I've got McArdle's disease and I decide I'm going to go for a run, what's going to happen? What's the first thing that's going to happen? I'm going to see my ATP levels fall, right? So I am going to kick in glycolysis so I can get my ATP levels back up. Glycolysis is going to use available glucose that I've got in my cell. Creatine phosphate is going to try to make up the ATP deficiency, and I'll have that, right? What's going to happen to my glycogen? Glycogen is just going to sit there. Nothing's going to happen to it because it's not going to be broken down by the phosphorylase. And since glycogen is one of the main sources of glucose for the cell, again, this seems very confusing and surprising. How does the cell survive? How is it that this person survives when they have the inability to break down glycogen in their muscle cells? We say, well, you don't exercise heavily. Well, just exercising is going to cause some need for glucose. This person, after they started exercising, would actually start feeling okay. There's a very important reason why. Yes, Robin? Yeah, the Cori cycle. The Cori cycle is what saves this person. So the glucose is coming from the liver. Once the liver gets the sense that glucose levels in the bloodstream are dropping, the liver, which has a normal phosphorylase, can start breaking down glycogen and supplying liver back out to the bloodstream, and the muscles pick it up. So McArdle's disease is a very surprising disease. Okay? It's very surprising in the sense that you're lacking one of the most important enzymes, but because of the Cori cycle, you actually survive the overall process. Yes, Lori? Why don't you get too much glycogen in the muscle cells? That's a very good question, and I don't know the answer to that. Very good question. You would think after a while, when you're sitting around, it's going to accumulate, right? Yeah. My suspicion is that, to maybe not totally cop out on the answer to that question, my suspicion is that the people who have this disease tend to be fairly low in blood glucose anyway, and so they become much more dependent upon Cori cycle. So they probably don't ever have situations where in their cells they have an excess of glucose where they can make much glycogen. That would be my guess, but it's a good question. I don't know the answer. Other questions? Okay. Well, I said I had a surprise, and one surprise was that I had a joke and something else, and the something else is coming up. So you can put your pens away and take out a piece of paper and pick up your pen. It is a pop quiz, actually, so. No, it's not. I'm just joking. All right, two surprises. There's two surprises. You don't need a piece of paper, okay? Oh, man.
I was hoping for extra points.
Extra points. Sorry, I don't have that. Okay. Well, what I do have is at the end of every term, you guys have been hearing me sing songs during the term, but at the end of every term, I write new songs. And so I've done that again this year. And I like to give the new songs to somebody who can actually sing them instead of me, because I, as you know, don't sing them very well. So I want to think about music. Music in biochemistry is very important. Biochemistry starts with a B, and B's are very important musically. Okay, you think I'm kidding? Look at this. Okay, the Beatles starts with a B, right? Okay, so that's a really important group, and maybe you guys have heard of the Beatles, right? You've heard of the Beatles? Okay. Well, the Beatles are really an important group. So I think that. Well, how did that get there? Okay. And after the Beatles, we have the Bee Gees to blame for inflicting disco on us. And they had a B in their name, right? And then after the Bee Gees, we had another group that inflicted all kinds of stuff on us. And they had two B's in their name. And then there was, we don't want to think about her. We can think of what she's inflicted upon us. Okay? So that brings us to the most important group of B's. The biochemical choir, who will be joining me on stage. And you can see that they have. So let me invite them to come to the front, the biochemical choir, which consists, if I can see who all is here, of Linda Benson, Lisa Shepard, Christine Schneider, and we're missing one of our choir. Okay, we have at least one more coming. Maybe he will show up as we get going. They're going to be singing some songs from their new hit album, Oops, We're Going to Do It Again. So what we have for you are several songs. The first one actually is not a new one. The first one is an old song that we've done before, but it's important because it's about the subject that we've been talking about a lot. It's called The Tao of Hormones. Okay? Please feel free to join us with this. Maybe the louder you sing, who knows, maybe the easier the exam will be. Listen to the tune of an old Simon & Garfunkel song called The Sound of Silence, and it goes as follows. Chemistry, my friend, it's time to study you again. Mechanisms that I need to know are the things that really stress me so. Get these pathways planted firmly in your head, Ahern said. Let's start with epinephrine. Membrane proteins are well known, changed on binding this hormone. Rearranging cells without protest, stimulating a G-alpha S to go open up and displace its GDP with GTP because of epinephrine. I can't hear you. Active G then moves away, stimulating at cyclase. So a bunch of cyclic AMP binds to kinase and then sets it free. All the active sites of the kinases await triphosphate because of epinephrine. Muscles are affected then, breaking down their glycogen. So they get a wad of energy in the form of lots of G1P. And the synthases that could make a glucose chain all refrain because of epinephrine. Now I've reached the pathway end, going from adrenaline. Here's a trick I learned to get it right, linking memory to flight or fright. So the mechanism that's the source of anxious fears reappears when I make epinephrine. Okay. Yes. Okay. All right. This is an old song probably none of you know, but I know, and Dr. Karplus knows, and Lisa knows, and Linda knows. So we're going to sing it for you. And you can join us. It's an old song called Close to You, sung by the Carpenters. The original tune goes something like, why do birds suddenly appear every time you are near? Okay. I'm not going to start. So I'll have you guys start, because I get off the wrong key when I start. So I'll let you guys do this. Enzymes truly are inclined to convert things they bind. Turn the key covalently, catalyze. How do cells regulate these roles? Allosteric controls to form C states R and T. Modulate. Competing inhibition keeps the substrates from the active site. They raise Km but leave Emax and shirk, while the non-competers bind elsewhere and lift the plot made on Lineweaver-Burke. Other ways enzymes can be blocked. When things bind, then get locked. Stuck, not free. Tied to the key, suicide. Idoglycan cross-links in bacterial cell walls in awesome ways. Beta-lactam rings reactive site, starts bonding with DD transpeptidase. So there are several enzyme states counteracting substrates. Now you see, blocking the key regulates. Catalysts have to be controlled. Some get slowed, put on hold. It's sublime how the enzymes catalyze. Okay. I screwed that one up, and I always screw it up. These guys know what they're doing, and I don't. So, all right, one more new song, and then we have one old one to finish. So, this is to the tune of "Coke, It's the Real Thing." If you remember that song, ready? I'd like to teach my students about some biochemistry to keep them all from drinking Coke and make them trans fat free. There'd be no taking creatine to help them run so fast. And sucralose and NutraSweet would be things of the past. I'd lower fructose levels in the junk that people eat. And salmon farmed and colored up, I'd work hard to defeat. Organic food is good for you, everybody knows. So let's get rid of factory farms and all the GMOs. Give us real things, some for you, some for me. Biochem's good, you see. Give us real things. Okay, and finally we have one oldie but goodie to finish us out. And this one is to the tune of the Yellow Rose of Texas. It's really easy to sing, and I really want to hear you on this one, okay? The term is almost at an end, 10 weeks since it began. I worried how my grade was 'cause I did not have a plan. The first exam went not so well, I got a 63. 'Twas just about the average score in biochemistry. I buckled down the second time, did not sow my wild oats. I downloaded the videos and took a ton of notes. I learned about free energy and delta G naught prime. My score increased by 7 points, a C+ grade was mine. I sang the songs I memorized, I played the MP3s. I learned the citrate cycle and I counted ATPs. I had electron transport down in all of complex V. I gasped when I saw my exam, it was a 93. So heading to the final stretch, crammed my memory and came to class on sunny days for quizzing comedy. I packed a card with info and my brain almost burned out. 'Twas much to my delight I got the A I dreamed about. So here's the moral of the song, it doesn't pay to stew. If scores are not quite what you want and you don't have a clue, the answers get into your head when you know what to do. Watch videos, read highlights, and review, review, review. Okay. That's all we have. Thanks to Linda Benson, Lisa Shepard, Christine Schneider, and Dr. Andy Carprice. Exam on Wednesday at 6 p.m. See you guys then.
Thanks, that was a nice surprise.
Yeah, good.
Smile for you.
Laverna. Wait.
I thought you did well.
Hey, Vicki, how you doing?
Hey.
Did you sing? Yes. Good. Hey, guys, good to see you. Glad you could make it. Well, as a matter of fact, look what I brought here for you.
I have present for you, Kevin.
Oh, thank you, Linda. Thank you. For all the goodie, for everybody.
Oh my God.
Enjoy.
You're so sweet. Thank you so much.
You're more than welcome. Thank you for coming. How you doing, Kiddo?
The new ones were wonderful.
Oh, good. They're fun.
And some,
I screwed the one up terribly. I always do that.
it just
It makes it funnier.
fun. It's all fun. It's all fun. Oh, there's a
Are the note cards up front?